Hi, I would like to thank the organizers for this great opportunity to present our work. I'm going to introduce two cancer genomics data resources we have been building, the cBioPortal for cancer genomics and OncoKB. I have nothing to disclose. In the past 15 years, cancer genomics sequencing projects such as the Cancer Genome Atlas and many institutional clinical sequencing efforts have generated a tremendous amount of cancer genomics data. The complex, multi-dimensional data sets imposed a great challenge for data access and analysis. To address this challenge, our group has been building software solutions in the past decade to make the complex data more accessible and interpretable, especially for cancer biologists and clinicians without computational skills. The two pillars of the data infrastructure we have been building are cBioPortal and OncoKB. We have integrated vast amount of cancer genomics data from various sources, including published data in the literature, Cancer Genomics Consortium such as TCGA and ICGC, institutional sequencing efforts such as MSK IMPACT, CCLE, and COMME-IN. In the public cBioPortal, there are currently more than 200 public studies. The supported molecular data include mutations, fusions, copy number, DNA methylation, RNA expression, and protein level. When available, we also include clinical data such as demographics, disease information, survival data, as well as longitudinal data such as treatment history. With this wealth of data integrated in our database, we spent a lot of time and effort to design and build intuitive interfaces for biologists and clinicians to query, visualize, and analyze this multidimensional data from various angles and make their biological and clinical discoveries. On top of the genomics datasets, we also connect to the various existing annotation resources and utilize them for our users to interpret the genomics events in patients. These resources include background biological data such as PFAM, PDB, UniProt, biological and clinical implications of variants such as OncoKB, CIVIC, My Cancer Genome, and ClinVar. We also include variant prevalence resources such as COSMIC, Cancer Hotspots, dbSNP, and NOMAD, as well as functional effect prediction tools such as SIFT, Polyphon, and Mutation Assessor. Among the annotation resources mentioned above, OncoKB provides the deepest integration with cBioPortal. In this talk, I will give a general introduction to both cBioPortal and OncoKB, and then do a demonstration of the data and integrative features provided by both tools. First, it's important to note the differences between cBioPortal and OncoKB. They are two standalone resources hosted on different websites. While cBioPortal is a cancer genomics database containing genomics and clinical data in cancer patients, OncoKB is a cancer variant knowledge base containing manually curated knowledge about cancer variants, but no patient data is included in OncoKB. The cBioPortal website provides a rich set of features for exploring, visualizing, and analyzing cancer genomics data, while OncoKB website provides functionalities for search and browse the curated information. Both websites are equipped with a high-performance web API. The cBioPortal API is for programmatic access to the data, and the OncoKB API is for variant annotation. The cBioPortal is completely open source and open access, while OncoKB API access requires a license, which is free for research use in academics. As I mentioned, the cBioPortal is completely open source. The code is available on GitHub with a GPL license. Our curated data files are also open source in the Data Hub. The software is now actively developed by the cBioPortal Development Consortium, consisting of multiple institutions, including Memorial Sloan Kettering, Dana-Farber, Princess Margaret, the Children's Hospital of Philadelphia, and the Hive, a company providing services around open source bioinformatics software. Besides the public cBioPortal, the software is also installed in many hospitals and academic and government research institutions, and also commercial companies. I highlighted the instances below installed by a few consortia here. Notably, the instance for the AACR project Genie contained 80,000 clinically sequenced samples from 17 cancer hospitals. You can sign up on genie.cbioportal.org for free access. The cBioPortal has been a popular resource for the cancer genomics community. There are about 4,000 visits per day to the public cBioPortal. Last year, we hosted more than 1 million visits. Our paper has been cited more than 6,000 times since 2012. We also have a poster in this meeting on cBioPortal. This slide is a quick overview of the poster. The cBioPortal interface is comprised of 4 main components: query result page, study view, group comparison page, and patient view. The poster will explain some more details, and I will also demonstrate these pages. Now I'm going to switch gear to introduce OncoKB. OncoKB is a precision oncology knowledge base that contains knowledge about cancer variants. It was created to address the challenge to interpret the vast amount of cancer variants that can occur in individual patients. When it comes to interpreting a variant in a cancer patient, there are 2 primary questions we try to answer. First, is the variant a driver mutation that contributed to the disease initiation and the progression? Second, is it clinically actionable? Or in other words, are there any drugs based on this variant? Or are there clinical trials that the patient can be enrolled? The OncoKB content is curated manually from various sources. The variant lists were retrieved from multiple variant databases, including cBioPortal, Cancer Hotspots, MSK IMPACT cohort, Cosmic, etc. Then for each variant, our curation team performed a comprehensive review of information from various sources, including FDA labels, NCCN guidelines, conference abstracts, clinical trials, and scientific literature, and synthesize the information based on the OncoKB curation SOP. The curated clinical evidence will then be reviewed by our clinical leadership to reflect MSKCC's best practice. Here is a summary of the curated content. Information on genes include the gene's role in cancer, such as whether it's an oncogene or tumor suppressor. Variant information including its biological effect, whether it's gain or loss of function, whether it's oncogenic, and clinical evidence, whether it's a biomarker to predict drug sensitivity or resistance. References for all evidences are also curated and available on our website. The data is available through an API. For the therapeutic evidences, we define a level of evidence. For the level 1 to 4, for drug sensitivities, and levels R1 and R2 are for drug resistance. The levels are based on the levels of actionability. For example, a level 1 evidence is an FDA-recognized biomarker that is predictive of response to FDA-approved drugs in the same cancer type. Level 2 is recommended by NCCN or other expert panels. Level 3A is compelling clinical evidence, usually based on ongoing large-scale clinical trials. Level 4 is preclinical evidence. In the past several years, we see expansion of biomarkers to targeted therapies, which we are able to capture in OncoKB. I'd like to mention that our levels are in agreement with the AMP, ASCO, and CAP levels. In addition to therapeutic implications, we are currently developing the levels for diagnostic and prognostic implications and associated data, and they will be released later. The information from various sources and the levels of evidence are curated and organized in a hierarchical format, from gene alteration tumor type to therapeutic implications. For example, BRAF V300E in melanoma and the lung is level 1 for these drug combos. But in hairy cell leukemia, it's level 2 for vemurafenib. The K601E in melanoma is level 3 for trametinib. OncoKB is powered by the clinical expertise of MSK. The content is curated by our clinical and research fellows, heavily vetted by a panel of senior physicians across MSK disease management teams, and the content is managed by a team of full-time PhD-level content manager and scientists. The OncoKB content is publicly available on oncokb.org, including information on 675 genes, over 5,000 alterations, 52 tumor types, and 92 drugs. All the information is searchable and browsable. Based on the curated content, we have also built a performant web API for programmatic annotation of cancer variants. The API is currently being used by the cBioPortal for annotating the variant in its database. To access the OncoKB API, a license will be needed and can be obtained on our website. The license is free for research use in academic setting. A fee is required for commercial setting or patient care setting. The OncoKB API is also being used by MSK to annotate the molecular reports of over 10,000 patients per year sequenced by MSK IMPACT sequencing panel. For example, this is a MSK IMPACT report for a non-small cell lung cancer patient. The patient has a number of somatic mutations and copy number alterations. OncoKB annotated each alteration to indicate whether it's oncogenic and whether there's a therapeutic implication. In this case, the tumor harbors 3 oncogenic mutations in EGFR, KEAP1, and TET2, and 3 copy number alterations. One of them is also in EGFR. The EGFR mutation has a level 1 drug association, and the M2 gain event has a level 3B association. For each drug association, a text summary is also provided in the report. OncoKB is also very useful for studying actionability in a cohort. We are automatically annotating all MSK IMPACT patients nightly, currently more than 50,000, and provide a report of actionability per cancer type. For example, from this figure, nearly 80% of GIST patients have level 1 drugs. Now I'm going to do a demonstration of both sides. First, I'm going to show OncoKB very quickly. So you can basically search for genes, alterations, tumor types, drugs, and also browse them. I'm going to show the example to search for gene. So for BRAF, you can load up the BRAF page and look at the gene summary and the background, and as well as the list of clinically actionable alterations, including tumor type, drugs, and level association, and as well as the PMIDs. And also you can look at all annotated and alterations, including whether it's oncogenic, gain of function, or loss of function, and their publication as well. You can look at other pages such as the levels of evidence, actionable genes, the cancer genes page, the API access page. This will require you to get a license, and there are other functionality. You can go to the About page. There's a OncoKB tutorial which will give you a tool detail of the details. For example, I'm going to use the cBioPortal public website. The public cBioPortal contains 285 cancer studies. We see a list of pan-cancer studies and a list of pediatric cancer studies and other studies organized per tissue type. So for example, this example, we're going to select the MSK IMPACT clinical sequencing cohort, query by genes. We're going to query our mutations and copy number events for EGFR. So once you submit the query, you'll see a list of tabs or analysis available. In this example, I'm going to explain OncoPrint and a couple of other tabs. So as you can see from here, 80% of all the cases in this cohort contain EGFR mutations of different types, fusions, or copy number amplifications or deletions. It's important to know that we separate the mutations into putative drivers and unknown variant of unknown significance based on OncoKB annotation, which can be changed here. Can also load clinical information. So, for example, I'm going to load the number of mutations per sample. It's interesting to know that if you look at the cases with EGFR VUSs, they have a much higher mutational burden here, indicating that this mutation is possibly passenger mutations. So the next one is cancer type summary. This tab provides a summary of alteration frequencies across different cancer types. And you see from here, glioma, non-small cell lung cancer, breast, sarcoma have higher alteration frequency in EGFR. The mutations tab gives a lot more detailed information about those mutations, and these mutations are plotted in this so-called lollipop plot. It's basically the number of mutations versus the amino acids, and from here you see that there are a couple of mutational hotspots in this cohort. And you can load additional annotations, including cancer hotspot, OncoKB, post-translational modifications, and 3D structure. So, for example, you can actually hover over and you'll see more information. For the 3D structure, you can also select the different alignments. And from here, what you see is basically a 3D structure in the p-kinase domain. Hover over all those mutations, you actually also see them highlighted in the 3D structure. The table down below gives you more information in those different samples, and this table is fully searchable. For example, I can search lung, and you see all the lung cases with the EGFR mutation, and you see a lot of mutations in the p-kinase domain. But if I search for glioblastoma, you'll see that a lot of mutations tend to occur in the extracellular part of the protein. I like to look at the annotations column more, and as I mentioned earlier, to help interpret mutations, we include annotation sources, including OncoKB, CIVIC, Cancer Genome, and cancer hotspots. And hover over will give you more information. For example, for the OncoKB, we will see that EGFR T790M is likely to be a oncogenic mutation, and it is known to be resistant to the first generation TKIs, and it is sensitive to osimertinib. For the second example, I'm going to use the AACR project Genie cBioPortal. If you haven't used it, you can register at genie.cbioportal.org. Once you sign in, you'll see that there are 2 studies, and I'm going to use the main cohort. So I'm going to explore the study this time. So this page will load the clinical attribute and genomics attribute for the almost 80,000 samples. From here, you see that non-small cell lung cancer, we have about 12,000 cases, and then followed by breast and colorectal. And there are a lot of clinical attributes, including sex, ethnicity, sample type, which is a primary or metastasis, the 15 centers, etc. And also now the we loaded the genomics data, so there are mutations, fusions, and copy number alterations. The top one mutated gene is a TP53. This page is fully interactive, so we can select, for example, the non-small cell lung cancer, and you see that the different the cancer type details, and also you see the top mutated genes for non-small cell lung cancer, fusions, copy number. So once we select the cohort, then we can, select more genes for more analysis. For example, I can select a couple of altered genes and then query this cohort for these genes to look at the details. So once it's loaded, you'll see that basically this is the same view as we demoed last time, and the idea is really to use the study view page to build your cohort and then use that to query genes for more details. For example, I'm going to introduce a new feature we have been building in the last year called a group comparison. So I'm going to switch back to the public portal again. And I'm going to select all the prostate cancer studies and then load them into the study view page. I like to compare the primary samples versus the metastatic samples for all of those studies together. So you see that there's a sample type attribute. Some of the samples are primary, some of them are mets. And from here, there's a button called compare groups. So it's basically comparing the primary and the metastatic samples. And from here, we see that there are two groups defined, A metastatic and B primary. We can actually swap them if you like. And then we'll be able to look at different data. For example, the survival data, we'll see that the primary samples, those patients have better survival, overall survival, as well as disease-free survival. We can look at the mutations. So this may take a little bit of time because this will compare for every single gene. It will compare the primary and the metastatic samples and see if any genes have enriched mutations in either primary or metastatic group. For example, we see that TP53, AR, MUC4, etc., they are enriched in the metastatic samples comparing to primary samples. The last example I like to show is the patient view page. The patient view page is a summary of all the clinical and the genomics data in a patient, and you can get to the patient view page from various places such as OncoPrint, Mutations tab, or the study view page. And for this patient, it's male, 22 years old, glioma. There are 4 samples from this patient, and if we have the timeline, you will be able to see what are the exact time points that those tumors get extracted, what are the treatment available in between. And also, all the mutations are listed for this patient, and you can sort them so that you know which are the new mutations, for example, in the recurrent tumors. We also, similar to the mutations tab, you see all the different annotations, for example, the OncoKB annotations here. So due to time limit, I'm not going to be able to explore more features in the cBioPortal. Please come to our website, and there are a lot of information in the tutorials and the recent webinars. Here is a list of resources that can be useful when using both sites. We also have posters for both cBioPortal and OncoKB. Please check them out. I will take some questions after this presentation, but for cBioPortal, if you have additional questions, please register for the live QA session with the cBioPortal team at 11 a.m. this Thursday. Please also send your questions to our user support group. Lastly, I would like to acknowledge all the contributors to cBioPortal and OncoKB. I was not able to combine both projects into one slide, so this is for OncoKB. Devyani Chakravarti is the lead scientist of OncoKB, overseeing the development of OncoKB. Nikki Schutz is the lead of our group, overseeing both OncoKB and cBioPortal. The rest of the team include the content and software development team, current curators, and the clinical genomics annotation committee representing the clinical expertise at MSK. Here is the multi-institutional team of cBioPortal. For the interest of time, I'm not going to name everyone, but mention that it is truly a marvelous team that I enjoyed working with every day. I would like to thank our current and previous funding agencies for supporting our project. Thank you for your attention.